Hi, it's Dr. Greg Mendel, your friendly anatomy professor. It is currently 2:22 a.m. and we left off on the first video talking about bone markings. And so what we said is that mostly bone markings is things you're going to look at in lab. So let's take just a quick peek here. But when you look at bone markings, basically the reason that we have things that are projecting off of the bone are for muscles to attach to have something to grab and to pull. And sometimes you'll see holes in bones, specifically like you'll see it mostly in a skull, and that's for things such as arteries and nerves to be going through, veins, that sort of thing. And you might see little grooves. And so these all have different names, like fossas and foramen and processes. And so you're just going to have to kind of look at the list because there's quite a long list. Tuberosity is an outpouching. There's a radial tuberosity where the bicep is attaching to. Long-headed bicep crest is like a raised area. Trochanter is a very large outpouching where muscles are attaching. And there's usually several attaching there. Line, again, is similar to a crest. It's a raised area. Tubercles are, again, very similar to tuberosities. Epicondyles is very similar again. It's just an outpouching. It depends on where it is and how to name it. Spines and processes are basically the same thing. The very outside, for example, head of the femur, head of the, different bones have heads. This is the head of the rib. Ramus means arm-like bar. We have one on our jaw, so on the mandible, mandibular rami. There's two different rami on the pelvis, and it means arm-like bar. So different little bumps and outpouchings all have different names. Sinus means a hollowed out area. So anyway, you're going to have to just look through the list, and when you start looking at different names of the bones, you'll start recognizing those. There are 5 major cell types that make up bone. Osteogenic means basically a beginning cell, very similar to like a stem type cell that's going to change and differentiate into other cells. Osteoblasts build bone and osteocytes maintain. The difference between an osteoblast and an osteocyte is simply location and age. As the osteoblast is a cell that secretes around itself, once it cements itself in, it's called an osteocyte. Bone lining cells are some on the outside, and osteoclasts break down bone. They actually eat bone. So osteogenic cells, again, sometimes they call them like a stem cell, sometimes they call them osteopropriator cells, but basically it's a active cell that can differentiate into an osteoblast or a different type of cell. Some just stay as the same stem cell. An osteoblast, again, builds. It secretes this goo very similar to the consistency of toothpaste, believe it or not. It's not hard like you'd think. It's secreting a goo that is called osteoid, and it's very, very similar to toothpaste, if you would. And so later, about 2-3 days later, it starts to harden with hydroxyapatates, and those are calcium binding, also phosphorus binding proteins. And so collagen is one of the big things here, and that's making up 90% of the bone's protein. Again, are not hardened by this one, but we have collagen being the majority of the bone's protein. Osteoblasts, again, are very active. They are very fast-paced, very metabolically high rate type cell. So here we have a osteoblast. There's a stem cell and it differentiates into osteoblasts. The osteocyte, remember that osteoblast cements itself in space, and that space is called a lacuna. And so once they cement themselves in that space, they no longer divide and they just maintain, and it changes names. So, but anyway, so their job is to maintain that bone. And so if there's an injury, it can help to build that bone back during a fracture or something like that, stress and strain on the bone. They also help them respond to mechanical stimulus. Now, the weird thing about this is that osteocytes have dendrites, kind of little pieces hanging out, and they can actually sense mechanical stimulus because there's some sort of an electrical signal happening there. It sends the signal to an osteocyte. The osteocyte will therefore communicate with other cells. It says communicate information to osteoblasts, which again build, and osteoclasts, which help to break down. So bone modeling. And so what that means is if you have an area where the bone is under a lot of stress, for example, a muscle pulling on it, it will lay down bone on the area which bone is being put on stress. But if it becomes too wide, it'll eat away at the inside to kind of balance out the equation there. Bone lining cells, again, they're just laying along the outside of a cell, but they're believed to help maintain the bone matrix along with osteocytes. On the outside of a bone, they're called periosteal cells because remember the periosteum is on the outside of the bone, and on the inside they're called endosteal cells because the inside lining is the endosteum. The osteoclast breaks down. Now, I remember talking about lysosomes inside of a cell. So osteoclasts contain a lot of lysosomes, and so an osteoclast is actually kind of made to look like a suction cup. And on the body, it will suction cup onto the side of a bone and secrete its acids and enzymes that will break bone down. And the reason it has a suction cup is it doesn't want those acids and enzymes to leak out, and it wants to have those lysosomes secreting their digestive enzymes basically right there. So it says when they're active, suction cupped on the side of a bone, for instance, says the cells are located in depressions called resorption bays. The word resorption means to break apart. They have little ruffled borders, again, the edge of the suction cup that serves to increase the surface area for enzyme degradation of bone. Degradation means to break apart. It breaks apart bone. And so as the bone breaks apart, those calcium salts go by endocytosis into the osteoclast, and then across by transcytosis, and then releasing by exocytosis into the bloodstream the calcium salts. But it also helps, again, like I said, suction cup, sealing off the area from that matrix. And so here's the suction cup side. And when you know that that suction cup's to you, it's going to release acids by lysosomal activity and eat. Here's an osteocyte, those little feelers, if you would. And we'll talk about these in a little bit and how they fit into a little area in the cell called a canaliculi. And we'll bring that up later. So compact bone also has this name called lamellar bone, and it makes up rings called lamella. And so the osteon is a Greek column, if you would. It looks like that tree trunk appearance where it has the multiple rings. The tree itself would be the osteon, and the rings would be the rings of lamella. The osteon has another name called a Haversian system, and the central canal within it is also called the Haversian canal, where the vein, artery, and nerve lie. There's two different types of canals. You have the Haversian canal, which goes up and down the osteon, but there's also some that go sideways, and they will be called Volkmann's canals, and we'll talk about those later. Canaliculi is very important, and they look like little bitty cracks within the lamella, and we'll show you those. The reason for the canaliculi is so that nutrition can be passed from different layers, starting at the Haversian canal to the outside, which is in the dead center of the osteon, to the outside, and giving this outside parts of that osteon nutrition from the artery. Now, interstitial and circumferential lamella we'll cover in a little bit, but circumferential lamella means that on a bone, the very outside is a wrapping of bone around the outside of the entire bone to make it strong. And interstitial, well, if you get a bunch of Greek columns, and what I mean by that is basically they're round, there's going to be spaces in between round things. And between those spaces, there's still bone, and that's called interstitial lamella. They're like leftover pieces of old osteons. So an osteon, or Haversian system, is basically the main structural unit of compact bone. As we said before, it consists of cylinders that look like a Greek column that go parallel to the long axis of the bone. In other words, from the top to the bottom, it runs that direction, from one end of the bone towards the other, not midway to midway. So it acts as a little weight-bearing pillar. Again, Greek column, if you think of it that way. An osteon cylinder consists of rings. Now remember, like the tree has rings called lamella. And again, the lamellar bone. Lamella consists of collagen fibers running in different directions. And what that means is that if you were to look at the rings of a tree, so it says like one ring, two, three, and there's the Haversian canal. But this one kind of goes clockwise, and that goes counterclockwise. And the next one goes clockwise. And that's what it says by they run in different directions. Now, the reason for that is a twistor resistor withstands stress and resists twisting. Bone salts are found between the collagen fibers. Now, collagen fibers we saw over here, where we said that basically the majority of the bone's protein is collagen. But we're going to harden that with calcium and phosphoric salts, which you mix those two together, they come up with a name called hydroxyapatates. We'll talk about that in a little bit, but those are the bone salts, hydroxyapatates. And so here's our twistor resistor, clockwise, counterclockwise. And so the central canal, which again is called the Haversian canal, goes through the dead center and has, as I said earlier, the vein, artery, and nerve. Now, they put blood vessels and nerve, but don't forget blood vessels are vein and artery. The perforating is the opposite direction, Volkmann's canals, which means it goes in right angles to the central canal. It goes back and forth instead of up-down. And so why? And that's because blood vessels can be connecting to other osteons, and it basically goes perpendicular instead of parallel with the bone. The lacuna is the space in which the osteocytes are in. Lacuna, space. And this little canaliculi are hair-like canals that the osteocytes' little legs can fit in. And I'll tell you a little story about canaliculi to help you understand it. Osteoblasts again build, and they secrete bone matrix when they're done, and they cement themselves in. They're called osteocytes. When the matrix hardens and the cells are trapped, the canaliculi form. Now, here's the way I'm going to explain this. Let's say we were in the classroom and I'm sitting in the front and I brought in a whole big pile of cheeseburgers. Now, those of you in the front row would say, oh, I want a cheeseburger, and I'd hand them to you, except people in the second, third, fourth, fifth, sixth, seventh rows also want one. So if you all imagine you turn around, everybody in the room would turn around and reach back to the row behind them to hand them a hamburger, your arms are the canaliculi, because they're the cracks in which the cheeseburger or nutrition in a cell can be passed from one row to the second, to the third, to the fourth, to the fifth. And the rows of desks inside a classroom are simply the circumferential lamella. And so it's one row, two row, three row, and any little crack in the middle is the canaliculi. You can't see them here, but I'll show you if you can. Now remember, that is in, and I'm going to go here, and that is in the osteon. This is an osteon, and I'm going to come back to this slide, don't worry. But this is an osteon, so you got the central canal or Haversian canal, first, second, third rows. But the canaliculi would be a little crack here. Now we're going to show a close-up of that here. Here's the Haversian canal. So there's the first row, second, third, and you can see the little cracks. And then inside that crack, the little extension of the osteocytes are passing food from the artery, glucose, to the bone back here, and then the cellular waste goes here. So you collect the cheeseburger wrappers into the vein and get it out of there. Now you can see there's a bunch of osteons throughout here. But again, as I said, if you got a bunch of circles, well, circles have a space in between them. So this little bone here is not an osteon, but it's filling in the gaps. It's between the circles, so that's interstitial lamella. And on the very outside of the bone, I said it wraps around 2, 3, 4, 5, 6, 7 times, and it goes all the way around the bone. That's the circumferential lamella because it circumducts the bone. Here we have the inside of the bone, which we know as the medullary cavity. And here we have some Haversian canals. And what do you know? A Volkmann's canal going perpendicular instead of parallel like the Haversian canals do. The very outside has periosteum, which is strongly connected to the bone by perforating or sharpish fibers. They perforate through into the bone. Here we have a microscopic view, by electron, I guess, of the central or Haversian canal. You can see rows of concentric lamella, and this is an osteon, if you would. Here again is an osteon. You can see there's multiple rows, rings, and the central canal. So the reason you're seeing a little hole here, little hole here, little hole here, little hole here, and those are lacuna in which an osteocyte would live. So the lacuna is the hole. The ring is the lamella. And the way I remember that is these little, I imagine a little osteocyte living in there. And I think it looked like a little bug, right? A little cootie and a lacuna. It rhymes. Lacootie, lacuna. And so the lamella is a ring for the rings of the tree. Spongy bone is not at all put together like an osteon. It is just haphazard speculations of stalactites and stalagmites, if you would. And so their trabeculae is like, it says, cables on a suspension bridge. They confer strength to bone, but it's basically these poorly organized, crazy, different directions of bone. It says they are, and you can see it in a femur head, and they line up along lines of stress to help make them stronger. But spongy bone, even if they're perfectly aligned, are not nearly as strong as a compact bone. There are no osteons in spongy bone, none. They're irregularly arranged lamella all over the place. There's capillaries and rich blood supply, and that's why red bone marrow is in the spongy bone, and this is where we make red bone marrow and bone growing blood cells. So blood is growing here. And so here we see spongy bone, here we see compact bone, but when we see spongy bone with a compact bone on each side, in other words, a spongy with a compact bone sandwich, that's called diplo. So here you can see it all kind of haphazard. The one thing they don't tell you is that this is someone with osteoporosis. Somebody looks at it often, would tell you that they would agree with me. This is osteoporotic bone. Actually, it's not extremely strong in this case anymore. And so bone is made up of both organic and inorganic compounds. And some of the organic ones, as we said before, the osteoblast, the osteocyte, the osteoclast, these are the living things, that are alive. But the osteoid is basically the goo secreted by the osteoblast. The osteoid says it's making up a third of the organic bone matrix, but we have to harden this. So there's something called the osteoid front or an osteoid seam. Somewhere around the 2-3 day mark, it starts to bring in these basically inorganic things called hydroxyapatates. And we talked about that earlier. We said they were mineral salts made up of phosphorus and calcium. So here we have the phosphoric crystals and calcium phosphate crystals. So it's a combination of a calcium and phosphorus. So they are responsible for the bone being hard and resilience to compression. Look at this. It makes up 65% of the mass of the bone, these calcium salts. And so what we see here is the inorganic things are half as strong as steel in compression and just as strong as steel in resisting tension. Wow. To think that your bone, if you're trying to squish it, it's half the strength of steel. But with tension, it's just as strong. That's wild. So they can last long after death because of the minerals in bone. And so that's why you see skeletons in graves, right? So they can reveal information about ancient people. They can do carbon dating. There's all kinds of things that you could do with bones and try to figure it out. Well, it looks like that was the remainder of the first half of the material for the chapter. And so I will be posting this also along with the other video. And ladies and gentlemen, I will finish the other video for next week over the weekend. And I hope you have a wonderful day.